Alright, SBI4U, we're into video number 4. I'm on a roll here, so I'm just going to jump right into it. Here's today's learning goals. First off, we're going to talk about, well, over there, free radicals and antioxidants. So you've probably heard of antioxidants and like superfoods and stuff, you know, blueberries and whatever. We're going to talk about what those are and why they're important. We're going to talk about uncoupling the electron transport chain with stuff called brown fat and also this drug called DNP. We're going to talk about the three different energy sources that humans can use depending on how much effort we need to put out. Then we're going to talk about fermentation versus anaerobic respiration. They're kind of related, but a lot of people get them twisted, so we're going to spend a bit of time talking about that. And then finally, a quick thing, anaerobic respiration itself and its use in bioremediation. So, jumping right into it. Let's pull up our current topic. The current topic is free radicals and antioxidants, and our keep in mind is that oxygen is really good at grabbing electrons from anywhere, from pretty much any chemical, any element except for fluorine. Fluorine is the only thing that's more electronegative than oxygen, the only thing that can make it, that can actually steal electrons from oxygen itself. So let's just jump right into the note. Let's talk about, okay, what's a free radical? Why do we need to eat antioxidants? Well, here's an example of a free radical down here. It's a compound that either hasn't fully bonded or has like an extra electron or something like that. So this guy here, you can see, is it's like an oxygen that's almost but not quite bonded. Jumping over to the document camera just for a quick second, oxygen, normally oxygen, the Lewis structure of it looks like this, where it's got two bonds and two electrons on each thing. But you can think of superoxide as, those two electrons are those two pairs of electrons are there. And this one has those two pairs of electrons. But this bond has kind of not formed yet, right? That bond hasn't formed yet. And it hasn't formed yet because there's an extra electron that has joined in. That's that red one in the picture. And because that's an extra electron that's joined in, that thing's negative. It would be great if, maybe another electron joined in here or something like that. Then you'd end up with a dioxide anion or something like that. This thing would love another electron, and that's why it oxidizes things. It's called superoxide. It's called a free radical because radicals are compounds that kind of either are missing an electron to be stable, or has like one, like remember bonds like being formed in pairs, so this thing's just got one, and it's going to grab an electron from somewhere. It'll grab it from basically whatever's available. It's not picky. Your cells actually use this stuff. Your white blood cells have an enzyme that makes this on demand. When they grab onto something, they will release superoxide in, and then this thing will just be like, right? Because that superoxide has grabbed up all the electrons and is messing up all the bonds in there. So when they grab like a bacterium or something, they pump this stuff into it. Now the bacteria and your cells as well can express an antioxidant. An antioxidant. In this case, it's a protein that's superoxide dismutase, which takes this and harmlessly renders it into like water and stuff like that, making it into, like, giving it some electrons, but electrons from the protein rather than from really important cell stuff. So here's like a kind of cute diagram of that. A free radical really wants to steal an electron off of something, and then the antioxidant's like, hey, I got a spare, it's cool. Right? I really like that diagram. This is the person that made it, some kind of pharmacology student that I guess likes to doodle. One thing that's particularly susceptible to free radical damage is DNA. That's because if you remember back in the biochemistry unit, DNA has a bunch of negative charge on it, so it's got extra electrons to give away. So DNA is particularly susceptible to free radical damage. And various things can make free radicals. So like sometimes radiation can come down and knock electrons away from something. And then now that thing is going to go looking for an electron from somewhere and might grab it from, say, DNA. Sometimes it's the white blood cells that are fighting something and some of this superoxide gets out. Sometimes, different like smoking and pollution and stuff, just toxins that you take in. And a lot of it actually comes straight from the mitochondria. So the mitochondria actually generate free radicals as part of their operation because their whole thing is moving electrons around, and sometimes it just hops the wrong way and suddenly you get a free radical. And as you get older, your mitochondria actually get less effective. Just like anything, they'll accumulate damage as they age. And I mean, they repair themselves and they replicate themselves, but you can't really stop it. And because they're making free radicals, the first thing that gets damaged a lot is the mitochondria too, because they're processing all this oxygen, and oxygen is great at grabbing electrons from anything, and you end up with more free radicals coming out, more DNA damage, and eventually, it starts doing damage to your cells that you can't repair. We do have, though, those enzymes and processes that kind of take the free radicals and turn them into harmless stuff. One of the major ones is vitamin E. This is vitamin E, or this is the general structure of a tocopherol, which is what the class of chemical that vitamin E is part of. And you can see here that this hydrogen can very easily pop off and get donated to the free radical. That dot is the extra electron, or the missing electron that the free radical needs. So the hydrogen just pops off and goes there. So in this case, right, the hydrogen would come in and join this guy, and just it'll just be an OH now, and it would be less damaging. This leaves this guy kind of like this, and then it can just grab a hydrogen from somewhere, some kind of hydrogen floating around, maybe off of a water. So, better than damaging your DNA. You're just damaging this vitamin E that you can then just get some more of in your diet instead of having to repair your DNA. So that's free radicals. And that's why antioxidants are good. Like you might have heard of people, eat blueberries, you'll age less and stuff like that. For like a really interesting example of this, I'm going to pause for a second and find this picture that I just thought of. Okay, yeah, so I found it. This is super cool. This went viral like a couple of years ago. It says, trucker accumulates skin damage on left side of his face after 28 years on the road. So a reminder that UV can actually generate free radicals just by hitting various atoms that are inside you, and then those atoms can go and damage your cells. Well, here's, the perfect example. He drove with the light hitting one side of his face for 28 years because he was next to the window on the driver's side, and this is what it looks like, right? This side of his face didn't get the UV. This side of his face took a bunch of cell damage, and you've got really good repair enzymes, but they're not perfect. So the skin on this side lost elasticity because I guess the collagen got affected, and then you can't repair it to the same degree as it had before. So yeah, UV will slowly, you'll accumulate cell damage from free radicals over time. This is one of the main theories of why we age, why like our skin gets saggy and stuff as we age. Still not 100% proven. This is one of those kind of unknowns in biology. What is the main kind of cause of aging? There's a bunch of different ideas. But we do know that preventing UV from hitting your skin will keep your skin looking healthier and younger. So use sunscreen as often as you can, and your skin will look younger because you won't accumulate that free radical damage. Basically, sunscreen works as antioxidants as well. The chemicals in sunscreen will give up their electrons, so they'll harmlessly absorb it instead of your tissues absorbing it. So that's pretty neat. That's free radicals. That is antioxidants. So we're good with part one. Next up, uncoupling the electron transport chain. So let's change our current topic to uncoupling the ETC. Uncoupling the ETC. And our keep in mind, I'll just save that. Our keep in mind is ATP synthase is not necessary for the ETC to keep going. Right? So ATP synthase is not necessary for the ETC to keep going. Okay, back to this, take that away, and moving on. So let's hack the ETC. Brown fat is something that babies have. They have it in their core and, kind of on their back behind, intrascapular it's called, and the scapula is your shoulder blades, so it's between the shoulder blades. And there's a little bit next to their kidneys. And what that fat is it's got an extra little protein in the electron transport chain that's called UCP1, which I think is literally just uncoupling protein 1, but that's probably wrong. And what it does is, it's kind of like ATP synthase, except it doesn't make ATP. It doesn't take ADP and make ATP. It just lets the hydrogen flow right back through, without making any energy whatsoever, or making any chemical energy whatsoever. And what that does is, you've got a bunch of hydrogen up here all trying to get through, and not a lot of hydrogen down here. As things start bumping around a lot, things start heating up. And so it just generates heat. Just starts generating heat. Adults retain some of it, but it's not the same amount as the baby. The baby needs this to stay warm, because they can't shiver. This is something that might be interesting to a bunch of you people watching. Babies can't shiver. They don't start shivering until later in life. Not that much later, but later in life. So that's why they need basically this like electric blanket inside them called brown fat to keep them warm. You see the same thing in hibernating animals, right? Just all the energy gets wasted as heat. Now there's another thing. There's a drug. It was actually started off life as an explosive in World War I. It was discovered that what it did is it could grab an H plus from the intermembrane space. So like, here's the mitochondria, here's the matrix, here's the intermembrane space where normally we have a lot of H plus building up, right? That's our H plus reservoir. It can grab an H plus from that intermembrane space and sneak it out of the mitochondria. Sneak it out and then release it and then come back in and then just do the same thing over and over and over again. So it effectively does the same thing. It lets this H plus go across the membrane without generating any ATP, and also releases it as heat. So very quickly it was marketed as a diet drug. People in the 30s like, oh, this is great. Why do you think it was a diet drug? There's a question. Why a diet drug? Why a diet drug? And I mean, you guys can pause the video while you think about that, but the answer is that you're using energy from glucose to pump the hydrogens up, but you're not getting any ATP on the way back. So yeah, it's pumping up, but you're not getting ATP on the way back. So you're processing glucose, you're processing carbs, you're processing protein and fats and stuff, but you're not seeing the energy going into your cells. So basically you're just burning calories for free, and literally burning them. You're getting warmer. So that was the problem. The drug, it was really easy to overdose on it and have too much uncoupling happen and too much heat happen, and people got really bad fevers, like really, really high fevers. And so it got banned in Canada in 1938, and it's banned around the world. But here's a recent headline. I think the start of this note, yeah, the start of this note had a recent headline where a banned slimming drug killed a medical student. You know, I guess she should have known better, but hey. And you can still buy this stuff. If you go online and just search for DNP, you'll find a bunch of sketchy websites that will sell it to you. Please don't. You'll probably get some kind of virus on your computer, but you can actually go and find websites that will sell it to you. They say, you know, for veterinary use only or for research use only, but I guess people are still into this stuff because it is such a powerful thing. Don't do it though. Like you'll get a horrible fever and you'll die. All right, next up. Oh, that was quick. Next up, 3 different energy sources that humans use. Specifically human muscles use. This I kind of stole from my physiology class that I've taught before. So let's talk about the 3 different energy sources. 3 different energy sources. 3 different energy sources. Used by muscles. Let's just add that to the thing. Used by muscles. And then up here in our keep in mind, all 3 are available all the time. All 3 of these are available all the time. All 3 sources, those are sources, are available all the time. So I'm going to give you general guidelines here, but it's not the end all and be all of this stuff. So first off, the kind of the big difference between these is how long they last and how much power they can give. So if you're 1 to 4 seconds or 4 to 10 seconds, you're using this ATP PC system, PC or CP. Different people do it different ways. And for the first 4 seconds, like if you're really just like really just a quick action, your muscles already have ATP ready to go, so you can just, and the ATP is already there. But that ATP very quickly runs out and only lasts 4 seconds. So you have this PC, which stands for phosphocreatine. Phosphocreatine, there's the name of it there, which is basically a chemical, a molecule that holds onto a phosphate, and it's a very high energy phosphate that very quickly will jump onto the ADP. So the creatine just gives up that phosphate, it sticks onto ADP. You get a new ATP right away, just one shot. Phosphate right onto the ATP. ATP can get used for muscle contraction. Do it again and again and again. So you can extend, your body can extend that like explosive power for a bit more time if it grabs that phosphate off of the creatine. And sometimes you'll see this used as a supplement for bodybuilders, right? Because they'll eat it. They'll eat creatine, they'll take a supplement, and then they want to make sure they have a lot of phosphocreatine in their muscles, because the way that you build muscle is lifting heavy weights, and if you can do more reps, use more of that explosive power for a little bit longer, you can get more muscle gain. So that's why they take creatine as a supplement. There's differing opinions on whether it actually helps or not, because your body can just make creatine on its own too. So yeah, it's not like a vital thing to get. But yeah, so that's your super quick burst energy, just like super fast. And this is unique to each muscle, right? It's not like a muscle in your leg could use creatine from your arm. It would have to get all the way to your leg in order to be useful. So each muscle has its own little kind of twitch explosive supply. But then that runs out. That runs out in 10 seconds. After 10 seconds, you still need ATP. You still need to contract your muscles. We're jumping over into anaerobic systems. So anaerobic means no oxygen. Anaerobic, like aero, right, is gas. So anaerobic means no oxygen. And what we mean for humans, what is anaerobic about this is it's just going through glycolysis. Glucose goes to pyruvate. You get 2 ATP. That's some quick energy for you. Not as quick as this, right? This is one shot. This takes 10 steps, so it's longer, but still pretty quick. and that goes off, and you can use that for muscle contraction. And your muscles actually store some glucose in them in the form of glycogen to do glycolysis too for some extra energy. And this can last about 2 minutes. With training, it can last longer. I've heard claims of up to 5 minutes. And you can run off of that without any oxygen whatsoever. Kind of a funny story. Back when sports science was a little bit new, they used to think that it was more effective to run while holding your breath. Like if you're doing a 100-meter dash, they thought that that would be the fastest way to do the 100-meter dash because you're not using energy in your lungs, breathing in and out, and so it keeps more energy for the rest of your body, and you're fine not breathing for 10 seconds. So people used to run the 100-meter dash holding their breath the entire time for like that 15 seconds or whatever it took to do the 100-meter dash. Now they do actually breathe because they worked out that that is faster. Don't ask me why. I'm not an exercise scientist, but I know that historically there was that time in history where everyone ran the 100-meter dash like this. It's kind of funny. Anyway, anaerobic. Now the thing about anaerobic is it's a little bit slower. It's 10 steps, and your muscles will very quickly lose power. Muscles quickly lose power during it. It's still, it's still a lot of energy. It's less than peak, but it's still a lot of energy. And this is used, for example, in the 400-meter dash, which takes about 40 seconds to do. This is like a strategy thing for the 400-meter dash that I found online. The first, little bit, you're accelerating up to full speed. Then here they call it cruise control. So the idea is to manage how much glycolysis you have to do, right? Because here, after 10 seconds, you're going to be out of phosphocreatine. But you're into the glycolysis. Then here they suggest starting to use the arms more to get more arm momentum, and then just full power down the straight, trying to kick up the oh god, I gotta run type system. Maybe trying to squeeze the last little bit of creatine energy out, to be like, oh, I'm getting chased by a tiger or whatever. That's kind of the idea behind actually having strategy for different races. That's actually why the 400 meter is my favorite race. The 100 meter is just, who's got the best creatine system, but the 400 meter, you have to balance the anaerobic stuff, the little bit of extra energy you can squeeze out of glycolysis. After that 2 minutes is up, you need ATP, and you need a lot of it, and so the only option is the oxidative phosphorylation, which we also call aerobic respiration. Oxidative actually refers to the chemistry concept of oxidation, but for our case, you can just think of it as aerobic, like you need oxygen for it. And then phosphorylation is the taking the phosphate, adding it to ADP, making ATP. So oxidative needs oxygen, phosphorylation, you're making ATP, and this only happens in the electron transport chain. And it requires oxygen, so the max power level of this stuff depends on how much oxygen you can get to those muscle cells at any given time. I should move this back. Oop, not that. That's not what I want to move. This is what I want to move. There we go. Yeah, so the maximum power level of oxidative phosphorylation of aerobic respiration is how much oxygen you can get to the cells. And this is why people do training when they're training for bike races and marathons and stuff. They're trying to get their lungs more efficient so that the lungs can pull in more oxygen. They're trying to get their heart more efficient so that the heart can push more oxygen to the muscle cells. And the muscles actually change how they interact with the blood in order to be able to have that longer duration energy release. So you can actually train your aerobic level quite a bit. Yeah, so that's your energy systems, and this tracks the time, the amount of time, and if you look up at the keep in mind, all three sources are available all the time. It's not like a clear cut, always this, always this, always this. You have all three of them available, and the preferred method changes. For example, if you're going for more than 5 minutes, you'll actually start burning like sugar and fat from other places in your body in order to try to help make more acetyl-CoA and that kind of stuff to get this going faster. Things like that. You've also always got a little bit of extra creatine, a little bit of extra stuff available to you just for like emergency power. Let's say you've been shoveling snow for a while and you're getting really tired, but then like a tree branch falls behind you, you still have enough energy to get that explosive ATP CP system getting into it. So yeah, we have different energy systems for different uses, and you guys have to know the difference between them, between the ATPPC, the anaerobic glycolysis, and the aerobic respiration. Please note, it's not anaerobic respiration. I know one of our things down here is anaerobic respiration. This is not anaerobic respiration. It's not anaerobic respiration. This is glycolysis, which is anaerobic glycolysis. Glycolysis is not respiration because there's no gas being exchanged. Back to our thing, back to our learning goals. So we've done the three different energy sources. Now we're into the difference between fermentation and anaerobic respiration. So take the goals away. And we are going to move on to fermentation versus anaerobic glycolysis. So it's a common point of confusion when students are learning this for the first time. This is that intermediate, between 10 seconds and 2 minutes energy source, it's designed to get quick energy, just this 2 ATP from the glucose. So 2 of them can come out pretty quickly. And then normally, in green here, what I've got is when you've got enough oxygen, in green is the pathway when there's enough oxygen, this NADH that's generated goes out to the mitochondria, it goes to the electron transport chain. The electrons get accepted by O2, and the NADH becomes NAD+, comes back here, and you can get the loop. So there's a loop here, right? Because you're constantly regenerating the NAD+. But if you don't have enough oxygen, if you've been running on anaerobic for too long, if you don't have enough oxygen to regenerate the NAD+, this stuff is going to build up. And you can't keep doing glycolysis because you're going to run out of NAD+. You're going to use it all up making NADH. So this is called oxygen debt. And you need to bring back your NAD+ before you can even get the quick energy. You won't even get this anymore because this is required as part of this step. So what does the body do? Well, it just changes into a different system where it takes the glucose and makes pyruvate, makes the ATP, right? That part, pyruvate. And then it can take the pyruvate and turn it into lactate or lactic acid. You might have heard of lactic acid. And that process regenerates the NAD+. So pyruvate turning into lactic acid is a way to regenerate the NAD+, and the NAD+ can continue the cycle and make 2 ATP each time. But you are completely missing out on the energy from the rest of the pyruvate. All those other 34 ATPs, if you send it to the mitochondria, you're just making lactic acid. That's what humans do. It builds up in the muscles, it gives you cramps and stuff, it makes your muscles sore. But sore muscles are a lot better than dying to the tiger that's chasing you. So you can run on this stuff for a while until you run out of capacity to store lactic acid. You've still got the oxygen debt after you're done. So this is why people breathe heavy after they finish a hard workout. You're trying to get the oxygen back, and the oxygen can actually turn the lactate back to pyruvate, and then that can go off to the mitochondria to do its work. Other organisms like yeast will use alcohol fermentation. If you look at these, they're very similar, very, very, very similar. Still the same idea. You want to regenerate the NAD plus, and just run off of your glucose there. So yeast does this in low oxygen environments as well. When it's got oxygen debt, it's going to go over to alcohol fermentation, and then we get to use that. We use that alcohol either to drink for fun, or we can put the yeast in with some sugar and other stuff, and it'll release CO2 and alcohol, and that'll puff up the thing that we've put it in and make bread dough, and the bread dough will rise because of the yeast releasing the CO2. And then we put it in the oven, we bake it, the alcohol burns off, and you end up with bread, which is pretty cool. So yeah, we kind of enslave yeast to do our bidding for us. That little fungus is very, very useful for us humans, and we specifically do it in a low oxygen environment so that it makes lots of this stuff. So the yeast goes over into like emergency power mode. Yeah, interesting application of metabolism. All right, last piece here, which is bioremediation using actual anaerobic respiration. So anaerobic respiration. So bioremediation. And anaerobic respiration is not lactic acid fermentation, is what we have to keep in mind up here. So in terms of a keep in mind, please note anaerobic respiration is not the same thing as lactic acid fermentation. Anaerobic respiration involves actually going through like a mitochondria type cycle of an actual like electron acceptor, but it just happens to not be oxygen. Lactic acid fermentation is that thing we just talked about where we're trying to regenerate our NAD+, not the same thing. So anaerobic respiration, our mitochondria and any mitochondria from like other animals or plants or yeast or whatever, they use oxygen as the final electron acceptor. But if you don't have oxygen, like for example, the bacteria that live in hydrothermal vents way down deep in the ocean where it's just like sulfur coming out, they might need to use a different thing. So, for example, they might use a metal ion like this iron, or they might use manganese or chromium or selenium or tellurium. But the ones that we're really interested in are like lead. Like lead is toxic. Arsenic's toxic. Mercury is toxic. Uranium is toxic. And so there are certain bacteria that we've identified and found that will actually process these heavy metals and make them less harmful as part of their electron transport chains, right? As the thing that accepts the electrons. Like for example, this uranium goes from 6+ to 4+, which means it had to accept some electrons to do that, right? It lost charge. So that's the concept of bioremediation. This is bioremediation. What you can do is you can actually take these guys, these specific microorganisms, and put them in the dirt, and they will chow down on these and turn them into less harmful versions of that metal, right? Take like an ion, turn it into a solid, as part of, like I said, as part of their electron transport chains. Some of them are bad, like the iron one will speed up rust in pipes and things like that. Sometimes when you see the really gross stuff coming out of pipes, it's actually down to those geobacter bacteria. But in other cases, we can use them for good, and that's bioremediation. So that's what bioremediation does and what it is. So back to the front, back to this, we have talked about free radicals and antioxidants and their role in aging, and how like UV and cancer, UV and smoking and stuff can increase the amount of free radicals, and how eating things high in antioxidants can reduce the amount of free radicals. We've talked about uncoupling the electron transport chain with brown fat for good in babies that can't shiver and in hibernating animals that need the heat, and for bad with DNP, that banned diet drug. We've talked about the 3 different energy sources, the ATP-PC system for burst energy, the anaerobic glycolysis system for like up to 3 minutes of kind of like, I just got to keep going here, but that creates lactic acid. And we talked about the aerobic respiration, which is oxygen, and you can go for hours on that stuff, but it's necessarily at a reduced rate because it depends on how much oxygen you can get to the muscles. Then we talked about fermentation. When you're running off of that medium system, off of the glycolysis system, you need to regenerate the NAD, and we do that by making lactic acid, which eventually cramps up our muscles. Then we talked about anaerobic respiration, which anaerobic respiration is using something that isn't oxygen to accept those electrons in your electron transport chain, and the idea of being able to use those bacteria to do good, to clean up toxic waste. That's it. Thanks for paying attention. Keep an eye out for the quiz, and I'll see you in class.